I am Craig Venter. I created goat mycoplasma using an African goat mycoplasma. I made the blue plague. The blue plague that I, Craig Venter, created eats the Gulf oil spill in 2010 and then creates poop biofilms to feed the molds and bacteria that are the type to infect humans off the Gulf or wherever the Gulf spill flows. I, Craig Venter, PhD, made a 6,000 base pair approximately 6 nanometers long. Mycoplasma, called Corexit or Synthea, a new form of microbiology, is not a natural biological organism. Genome scientist J. Craig Venter, the founder of Synthetic Genomics, Inc., is clearly defined as this new biological structure in his prepared testimony before the U.S. House of Representatives Committee on Energy and Commerce. One of the major advantages of synthetic genomics is that there is no need to have access to a physical supply of a particular DNA sequence. Sequence fragments are simply created de novo by chemical synthesis and assembled into entire chromosomes and organisms. This ability to synthesize DNA and use it in the construction of new cells can catalyze a major change on what organisms can be engineered to do. These synthetic genome technologies could be used to produce bioremediation techniques. In 2003, they successfully synthesized a small virus, approximately 6,000 base pairs long, that infects bacteria. The team was also able to synthesize a small bacterial genome. In Bridging the Gap radio interview with Dr. John Waterman on September of 2010, he stated, "Microbes can morph, they can change. Viruses can turn into bacteria, and bacteria can turn into fungi. In the Gulf War, we have bacteria that can morph. It can morph, mutate, because it's attacked by a virus. The virus can change the genetics of the bacteria so that it morphs and it mutates into something very deadly. Some of these changed bacteria can become deadly, Ebola deadly. When you have a morphed bacterium that gets airborne, now you're going to see it go from person to person. We're on the verge of something can become a deadly pandemic. They have to know that was the case. All it has to do is enter the human host, and once it gets started, it's going to be impossible to stop. So it was created using a yeast, then transferred into a bacteria that changed the bacteria to be self-replicating. This is the first synthetic organism. Its website is encoded into its DNA. It is chipped. He says he uses his technology 15 years prior to 2010 to encode Haemophilus influenza. In early 2008, scientists at the J. Craig Venter Institute announced that they had manufactured the entire genome of a bacterium by painstakingly stitching together its chemical components. By sequencing a genome, scientists can begin to custom design bootable organisms, creating biological robots that can produce from scratch chemical humans can use, such as biofuel. And in 2010, they announced they had created a synthetic life, DNA created digitally, inserted into a living bacterium and remaining alive. Obama likes it.